So welcome back from the break. Let's take a look at our concept check questions so we can keep moving on. So compare the replication of the leading strand and the lagging strand of a DNA molecule. Identify the different enzymes involved and their roles. So leading strand. Well, we could perhaps start before that. We've got helicase, unzips and unwinds the two strands. So that would be the first enzyme that we want to pay attention to. There is another enzyme involved that we haven't talked about, one called topoisomerase, that functions on the DNA ahead of the replication fork to prevent the DNA from twisting up into knots as it unwinds very, very rapidly, because that is a potential problem. And so the topoisomerase relieves the tension that builds up in the DNA as it unwinds very rapidly. We actually haven't talked about the rate of replication, but in a bacterial cell, the rate of replication is somewhere around 500 to 600 nucleotides per second are being added in. So you can imagine how quickly the DNA is being unwound under those circumstances. In eukaryotes, it's only about 50 to 100 nucleotides per second, much slower, but nonetheless quite rapid number. So the role of topoisomerase is quite important. So leading strand. Let's talk about the leading strand first. So the leading strand primed once, by primase, that's the enzyme, then continuously copied by DNA polymerase. Leading template. In the 3' to 5' direction. And remember again, the new strand is being built in the 5' to 3' direction. Okay, lagging strand. Where am I going to put that? I'll put that up here. Lagging strand. Continuously reprimed. Then copied in fragments. And these are the Okazaki fragments. Put a full stop there, I think. Let's get rid of this. Second, DNA polymerase. Removes primers and substitutes DNA nucleotides.
DNA ligase links the fragments.
So that gives you the basic idea of how each of the two strands are copied and the enzymes that are involved in the process. So let's now start taking a look at transcription and the extraction of information from the DNA molecule. So what is the role of the DNA? Well, it's a repository of information for producing proteins. Pretty much that's what it is. The primary product of most genes is a protein, or at least a polypeptide. Remember what we said about the relationship between proteins and polypeptides. So the product of a gene is a polypeptide, which may be a protein in its own right, or that polypeptide may have to interact with other polypeptides to form a protein. But the primary product of the vast majority of genes is a polypeptide of some description. It is the extraction of information out of the DNA molecule in the form of a messenger RNA molecule that allows for the synthesis of the polypeptides. So there is a distinct flow of information in the cell. Now, we've talked about genotypes and phenotypes before. We don't really need to review that, but just in case you've forgotten, the genotype is the gene makeup of the organism, the genetic combination that's found in the organism. The phenotype, of course, is what it actually looks like, and that is essentially the proteins and the result of those proteins being present. So there are two distinct steps, and we use language terms to describe these, transcription and translation, because we use the analogy of language to describe what is going on. When you transcribe something, you're basically changing from one form of a language to another, or you're making a copy of an existing form of information in a new form. So scribes practiced transcription. And so in the past, if you wanted a book, before there were printing presses, you might hire a scribe to make you a copy of an existing book. And the process of making that copy is called transcription. Or until about 15 years ago, all the information that a doctor would collect on a patient during the day, rather than being written down, would be recorded on a little dictaphone or something like that. And then in the evening, someone would sit down and listen to that and type out what the doctor had dictated on that dictaphone. And they were referred to as transcriptionists. They were changing the spoken language into written language. So you're not actually changing the language, you're just changing the form of the language. And in transcription, as far as cellular information is concerned, that's basically what you're doing. You're going from DNA, a polynucleotide, to RNA, also a polynucleotide. You're just changing the type of polynucleotide that you're using. So transcription is an appropriate word to use here. Translation in language means changing from one language to another. So you might change from English to Spanish, or Spanish to English, or English to Greek, or whatever two languages you want to refer to. You are changing from one language to another. We even talk about computer languages as a way of representing information. And sometimes we have to translate computer language to something that humans would understand, or we have to change what humans understand to something that a computer would understand in a computer language. We are translating from one language to another. And in translation in cells, in cellular information, you're changing from the language of nucleotides in the form of the RNA to the language of amino acids in the form of the polypeptides. So you really are changing from one language to the next, or one language to another. And it's all built around the notion that we recognize that cells speak these two distinct languages, the language of nucleotides and the language of amino acids. And somehow in the flow of information, you have to be able to not only make a copy of the original message, but also translate it into its new form. So this animation, again, is a really cool animation showing transcription and translation in process. So it will walk you through the process of transcription, making a copy of the information in the DNA molecule as a messenger RNA. It will run you through what happens in a eukaryote in the form of what is referred to as post-transcriptional processing. We'll talk a little bit about it. And then it will show you translation, where the ribosomes read the information in the messenger RNA and assemble a polypeptide by linking amino acids together based on the information in the messenger RNA, the process of translation. This is a really nice animation, about 3.5 minutes long, as all the BioFlix animations are. And it does a really nice job of showing you transcription and translation in process. So I'd strongly recommend that you take a look at this. And of course, it's in the resources page. This is our basic flow of information. This is sometimes referred to as the dogma of molecular biology, and it was actually first proposed by Francis Crick. And he basically said that the flow of information is DNA to RNA to protein. And in bacteria, it really is as simple as that. DNA to RNA to protein. In eukaryotes, it's a little bit more complicated by virtue of the fact that the layout of genes in eukaryotic DNA is a little bit more complicated than the simple way the genes are laid out in bacterial DNA, which means that the product of transcription cannot be used immediately by the ribosomes. It needs to be processed, hence the need for post-transcriptional processing of the product of transcription. But ultimately, what is finally recognized as a messenger RNA is passed out into the cytoplasm, where the ribosomes then translate that information into polypeptides by assembling amino acids together. So that's what we're going to look at. First transcription, then translation. So once we start recognizing that the cell is actually speaking these two languages, the language of nucleic acids and the language of amino acids, it also becomes necessary to think about how this process takes place and how that information as nucleotides can be converted to sequences of amino acids. Because remember again, if we look at the number of nucleotides present in DNA or the number of nucleotides present in RNA, there are only 4 nucleotides. Yes, they are a little bit different between RNA and DNA, but there are still fundamentally only 4 nucleotides. But when we start thinking in terms of amino acids, there are 20 amino acids that commonly are used to make polypeptides. There are a few others that we are aware of, but only 20 of them are universally used to make polypeptides amongst all organisms. So we have a fundamental problem. You've got 4 nucleotides, but 20 amino acids. How do you translate from 4 nucleotides to 20 amino acids? It obviously can't be a one-to-one equivalent. 1 nucleotide cannot be equivalent to 1 amino acid, because that would only allow us to code for 4 amino acids. It's got to be more complicated than that. But it doesn't take very long to figure out exactly what the coding ought to be. Because if you can't use single nucleotides, maybe you have to use combinations of nucleotides. So the smallest combination is 2 nucleotides. And given that you have 4 different nucleotides, there are in fact 16 possible combinations that you can make out of that. That's very close to what's needed. We need enough for 20 amino acids, but it's not enough. So it turns out that it can't be combinations of 2 nucleotides. Well, the next smallest combination of nucleotides, we could say words if you want to use language terms, is 3 nucleotides. And if you do that, then the possible number of combinations is 64, which is way more than we need. We only have 20 amino acids. Now we have 64 possible combinations. It turns out, however, that all of those 64 combinations have a use. It's what is called the genetic code. And it took about 12 years to figure out what all 64 possible combinations actually referred to. But after about 12 years, it was finally determined what each of those 64 3-nucleotide combinations, what we refer to as codons now, actually refer to. And we now have what is referred to as the genetic code. And we know that that genetic code, with the exception of a couple of very, very small exceptions, is truly universal, which means that any organism can read and act on any gene from any other organism because of the universality of this genetic code. It tells us that that's possible, but it also tells us that the origin of the genetic code itself must lie way back very, very early in the history of the evolution of life, because if all life uses essentially the same genetic code, then it must have been one of the first things to evolve as life itself started to evolve. This must be one of the very, very earliest features of life to evolve, because every life form uses exactly the same sequence of nucleotides in its genetic code. So here's the flow of information. From DNA, through the process of transcription, we create the messenger RNA. Then the sequence of nucleotides in groups of 3, known as codons, then are recognized by the ribosome and used to assemble amino acids following the sequence of codons, each codon representing a single amino acid, to create the coded polypeptide that is represented by the information in the messenger RNA. It sounds like a fairly simple process, and to all intents and purposes, it is relatively simple, but it does require a little bit of explanation. So we're going to dig into some more detail here and talk a little bit more about how each of these processes take place. So I just talked about the notion of a codon being a 3-nucleotide sequence of nucleotides, so we don't have to dig into that in more detail, that there are 64 triplets or codons. Let's just take a look at the genetic code. Now, in your book, there is what I think is a neater way of representing this. This is the circular diagram of the genetic code. It's a lot easier to read than this table form, but this is the traditional form of representing the genetic code. And if you look at the nucleotides that are present, you'll see that there are U's present. So that tells you that we're dealing with the information in the messenger RNA, which is, after all, what is going to be translated. So the codons are always represented as the sequence of nucleotides as you would find them in the messenger RNA. So we read this diagram by looking at this one first, then we come round and pick up this one, then we come round and pick up the one on this side. And what immediately stands out is that there are 3 codons that are dedicated to stop. After all, if you've reached the end of the information, you want to make sure that the ribosome gets the message, don't do any more assembly. So each of these codons represents a clear stop message. So when a ribosome encounters one of these three codons, it stops the process of translation and assembling polypeptides. Then there is another codon down here called the AUG codon, which also codes for methionine, that is referred to as the start codon. Because again, if all you've got is a sequence of letters, and that's basically what you've got as far as a gene is concerned, it's a sequence of nucleotides, so it may as well be understood as a sequence of letters. Unless you know how to break that up into groups of 3 nucleotides to form the codons, you basically can't read the message. So the first thing you need to do is recognize where the message begins. And so there is a single way of recognizing where the message begins, the AUG. That is always the start codon, which means that the first amino acid in the vast majority of polypeptides is a methionine, because AUG is the codon for methionine as well. Now you might think, well, that could be a little bit confusing. Which of the AUGs do you know is the start codon? Well, the messenger RNA contains the information for that, and it helps the ribosome recognize which AUG it should treat as the start codon. So all is not lost there. Then as we take a look at the other 61 codons, what we start to recognize is that many of the amino acids have multiple codons, and some of them have a lot. So serine, let me change the color on this for a minute. So serine here has these 4 plus these 2, so there are 6 in total. Arginine, do it in yellow, has these 2 plus these 4. And then the other one, fine, yeah, leucine's the other one. Leucine has these 4 plus these 2. And what you notice there is that the only difference between these 4, in fact, is the last letter. So long as CU is present, doesn't matter what the third letter is, you're always going to get a leucine. And so the way we describe this is to say that there is redundancy built into the genetic code. And as you go through and take a look at the other amino acids and the other codons, you'll find that every amino acid, with the exception of AUG, has at least 2 codons coding for it. I think that's correct, if I remember. I'm just casting, no, tryptophan, yeah. I had it back in my mind that one other amino acid had a single codon. Tryptophan has a single codon as well. So methionine and tryptophan have a single codon. All the other amino acids have at least 2. Many have 4, and these 3, leucine, serine, and arginine, have 6. But that's a reflection of the importance of those 3 amino acids in terms of the function of polypeptides. So not surprisingly, they have multiple codons to make sure that they get incorporated in the correct position. When you want one of these amino acids, you want to guarantee that you get that amino acid. And the same is true for many of the amino acids that have 4 codons as well. Again, it is the last nucleotide in the codon that is the difference. That's become known as the wobble base, meaning that it can wobble, it can vary without changing the nature of the signal that's being sent. As long as the first 2 letters are correct, doesn't matter what the third one is. It can be 1 or 4. You're still sending the same message. So there is redundancy here. But what you'll also notice is that every codon only codes for 1 amino acid. Even the AUG only codes for a single amino acid. None of the codons does double duty in terms of amino acid coding. Now, the AUG does double duty because it's not only the codon for methionine, it's also the start codon. But again, that doesn't change the fact that it's still coding for 1 amino acid and only 1 amino acid. So while there is redundancy, there are multiple codons for each amino acid. Each codon only codes for one amino acid. In other words, there's no ambiguity. No codon does double duty when it comes to coding for an amino acid. So there may be redundancy, there may be multiple codons for a particular amino acid, but every codon only ever codes for a single amino acid. There's no ambiguity. There may be redundancy, but no ambiguity. So these are the features of the genetic code. So here's an example of taking advantage of the universality of the genetic code. And I'm going to draw an arrow from this piggy down here. Because this pig contains a jellyfish gene. So this pig actually contains this gene for the protein called GFP, which stands for green fluorescent protein. Why does the pig need the green fluorescent protein? Well, it doesn't. But this gene and its resulting protein is used as a marker by genetic engineers so that they can tell that the DNA that they were trying to insert into the cell that carries the genes that they really want to get in there has actually gone in, because the presence of the GFP protein and the green glow indicates that the DNA went in, which is a pretty good indicator that the other genes went in as well. And so this pig has this protein in every one of its cells, and consequently all of its cells glow green as a consequence. And in fact, we have two other jellyfish proteins called red fluorescent protein and yellow fluorescent protein that are used for the same thing. And you might ask, why does a jellyfish need these? Well, these are jellyfish that live deep in the ocean where there's no natural light. And so they produce their own light through these fluorescent proteins that glow in the dark, which obviously has good points and bad points. If you glow in the dark, you might attract a mate, you might attract something to eat, but likewise, you attract things that want to eat you. So it's a trade-off. But we use these in genetic engineering exercises to act as markers so that we know that the pieces of DNA that we want to get in to the cells actually go in. The marker tells us that they're there. If you have aspirations of going into the nursing program and you eventually take our microbiology course, you'll get an opportunity to insert this protein into bacteria and create colonies of bacteria that glow green, just like this piggy does. It's very easy technology to work with. But again, it revolves around the fact that the genetic code is universal. Every organism uses exactly the same set of codons, and every organism understands the message from the genes because of the universality of that genetic code. So before we move on and actually talk about transcription in great detail, a quick concept check. This is not going to be a full break. We're just going to answer this concept check question and then move on. So how many nucleotides would be necessary to code for a polypeptide containing 100 amino acids? Well, the first thing that you need to do is remember how many nucleotides represents a codon for an amino acid. And if you think back, there are 3 nucleotides per codon. And of course, there's 1 codon per amino acid. So it's a simple math thing. We multiply the 3 nucleotides per codon by the number of amino acids, which is 100. And that tells us we would need 300 nucleotides. To code for a polypeptide with 100 amino acids. Pretty simple. So when we start thinking in terms of transcription, what we need to remember is that we are going to be using one of the 2 strands in the DNA molecule as the template for generating what is referred to as the transcript or the messenger RNA. So the first thing that we need to do is to separate the 2 strands, just as we saw in DNA replication. However, this time, because we're not making a complete copy of the DNA molecule, nor are we attempting to copy both strands, we only want to copy the template strand. What happens during transcription is a little bit different. So the first thing that we need to recognize, and this animation shows you the basic process of transcription. Oops, let's just clear that out and start again. So this animation just shows you the basic process of transcription. Nothing more complicated than that. And it works you through what happens in the transcription process. Now I'm going to talk about it in a little bit more detail. So I'm just going to move on, but you should take a look at this animation to get a basic feel for what transcription is all about. So there are 3 recognized stages to transcription. Initiation, where the enzyme that actually creates the transcript, called RNA polymerase, binds to a region of the DNA outside of the gene, but adjacent to the gene. Then the process of elongation is the basic process of creating the transcript or the messenger RNA. Then termination takes place when the transcription or the transcript is complete and there's no necessity to continue the copying process, because that would introduce information that is no longer relevant to the gene. So the RNA polymerase lets go and releases the transcript and also lets go of the DNA. So let's look at the process of initiation, elongation, and termination in a little bit more detail. So initiation begins at a region called the promoter. Now, remember again that the template strand is read, as we said previously, 3' to 5'. And this is referred to as so if you're moving 3' to 5', because that's the direction that DNA is always being read in, we say that that's going downstream. So the promoter is located upstream from the gene. Which obviously means going in the other direction, going 5' to 3', not 3' to 5'. So when the RNA polymerase binds to the DNA, it binds not at the start of the gene, but at the promoter, which is upstream from the gene. It is attracted to the promoter by a series of proteins called transcription factors that bind to an area of the promoter, and that provides a signal to the RNA polymerase that this is where it needs to bind to begin the transcription process. It then attaches to the promoter and starts moving downstream, in other words, starts moving 3' to 5', and it starts to separate the two DNA strands. But it only separates about 20 to 30 nucleotides, a very, very local little bubble. And it starts reading the template strand. That is the strand that goes 3' to 5', and starts assembling RNA nucleotides in the 5' to 3' direction. It doesn't need a primer in order to get started. It can immediately start assembling the transcript by reading the template strand. So it starts reading the template strand 3' to 5' and starts building the messenger RNA or the transcript by adding nucleotides 5' to 3' on the growing strand. Then the longest phase of transcription, elongation, is simply a continuing opening up of the DNA by the RNA polymerase and a continuous copying of the template strand. As the DNA exits the RNA polymerase, it immediately snaps back together and rewinds again because it no longer needs to remain open. So you only ever have this local small bubble of about 20 to 30 nucleotides max that are open as the RNA polymerase moves its way, reading the template 3' to 5' and assembling the transcript. Eventually, it comes to a sequence of nucleotides at the downstream end of the gene. They're actually outside the downstream end of the gene or the 5' end of the gene. And that sequence of nucleotides tells the RNA polymerase to stop. It signals that this represents the end of the gene, that transcription should end, and that the polymerase should release from the DNA, and that the transcript, the RNA molecule, should also be released. And as that happens, the DNA snaps together, the two nucleotide strands of the DNA snap back together again, and the DNA winds back up into the double helix and transcription is over. Now, if we were in a bacterium, if we were describing the bacterial situation, excuse me, the messenger RNA would be ready to be translated. There would be no further requirement for any processing. In fact, translation might have already begun before transcription has finished. Because remember, the DNA in a bacterium does not sit inside a nucleus. Everything takes place inside the cytoplasm of the cell. We have this special region in the bacterium called the nucleoid where the DNA is located, but there's no membrane to separate them. And so the ribosomes can actually attach to the messenger RNA before transcription has actually finished and begin translation. And this is part of the explanation for why bacteria can grow so quickly, because they get on with the process of translation even before transcription is finished. In a eukaryote there, however, that's not the case. The transcript is not the messenger RNA, and that's because of the nature of genes and the way they're laid out in eukaryotic DNA. And remember, of course, transcription is taking place in the nucleus, but translation is going to take place out in the cytoplasm. So before translation can even begin, the final messenger RNA has to exit the nucleus out into the cytoplasm. So there are some added complexities to the eukaryotic situation. So what happens here? Well, there are two things. We get to the diagram. So the first thing that needs to take place is that the ends of the RNA molecule need to be protected, because if they're not, out in the cell are enzymes that will immediately break down the messenger RNA. In fact, it's a way of regulating protein expression, which we'll see when we talk about that in the next chapter, chapter 11. So the first thing that happens is that a modified G nucleotide is added to the 5' end of the RNA. At the 3' end, a long sequence of A's are added, what's referred to as a poly-A tail. And the length of that tail is actually coded within the transcript and tells the cell how long to make the poly-A tail. And the length of that poly-A tail tells the cell how long to leave the messenger RNA alone before you break it down. And so the genetic information actually tells the cell how long to leave the messenger RNA intact by telling it how long to make the poly-A tail, which provides that signal. The other thing that needs to take place is that internally within the transcript, there are regions that do not contain coding information. These are referred to as introns. And this is something that is unique to eukaryotic genes. Eukaryotic genes are not laid out in a continuous fashion. In bacterial cells, they are. And that's the reason why the transcript is also ready to be translated, because it is the messenger RNA. It doesn't need any further processing because there are no introns in bacterial DNA, but there are in the genes of eukaryotic cells. So we need to cut out those introns and splice the rest of the transcript, what are known as exons, together. And so that takes place inside the nucleus. And there are complexes of RNAs and proteins called spliceosomes that carry this out. They cut out the introns and splice together the exons. And they can do this in a number of interesting ways in order to vary the product of the gene. We used to think that one gene gave rise to one polypeptide. That we don't think anymore, because we know that when the spliceosomes splice the exons together, they may not necessarily splice all the exons together. They may leave some out. And they may not necessarily splice the exons together in the order in which they occur in the transcript. So if I put numbers on, well, yeah, if I put numbers here and say that this is number 1, number 2, and number 3, they may not necessarily get spliced together in that order, 1, 2, 3. They may get spliced together in a different arrangement, 2, 3, 1, or 2, 1, 3, or 3, 2, 1, or 3, 1, 2. Any number of possible combinations can occur. And of course, if you splice the exons together in different orders, you change what sort of polypeptide you're going to make. And you might wonder where this might be a benefit. And where it might be a benefit is something that we're dealing with right now, our immune system. This is the reason why our immune system is so flexible and can make antibodies against the large range of pathogens that it's capable of making antibodies against. It only has a limited number of genes to play with, but it recognizes several times more pathogens than the number of genes that it has to play with. In fact, it's tens of times more pathogens. And the way we can explain this is that within the genes that make up the genes responsible for antibodies, there are lots of introns and exons. And when the introns are cut out and the exons are spliced together, the spliceosomes create multitudes of varieties of spliced exons that produce a variety of resulting polypeptides. When one of them starts working as an antibody against a new pathogen, that's the one that the cell remembers how to make. It forgets how to make all the others and just remembers how to make the one that works. And so your immune system creates a memory of how to make antibodies against every pathogen that your body is confronted with. And it does so by splicing exons together in different ways, in different combinations, until it finds one that works. So this is something that, again, that we'll talk about in Chapter 11 in a little bit more detail. But it's interesting to start talking about it here. I think it's relevant to start talking about it here because we're already talking about cutting out introns and splicing exons together. Once that splicing takes place, once we've got the cap and tail on, once we've done the splicing, then the messenger RNA is complete and intact and ready for export out to the cytoplasm to be translated by the ribosomes. So let's take another break. This time we will take a formal break. Here are the concept check questions that I'll answer for you when we come back from the break. What region identifies the start of a gene to the RNA polymerase? And in eukaryotes, the final mRNA is shorter than the original transcription product. Why? I'll see you on the other side of the break with the answers.